Hey everyone, Mr. Andy here, and I wanted to go over with you some information about cell division, particularly the cell cycle, and within the cell cycle, the process of mitosis. So let's think about our big questions here. So why do cells need to divide, and how does cell division provide for the continuity of life processes in an individual and in a species? So why divide? The simple answer is for the continuity of life. And so the metabolic processes that our bodies and all organisms' bodies carry out every single day damage cells. And cells have a good ability to repair themselves, but eventually those mechanisms wear out. And so an individual, in order to survive, has to replace the damaged cells that no longer work efficiently. Two, in order to grow, cell production has to be greater than cell death. And so on a species level, in order for a species to survive, the species has to replace the individuals who died. So cell division accomplishes all of those processes. So particularly, let's talk about what is the cell cycle. So the cell cycle refers to the phases of a cell's life. The majority of the time, a cell is going to be in what is called interphase, approximately 90% of a cell's life. That encompasses G1, which is the growth phase. So the cell, when it begins the cell cycle and enters into G1, will actually grow in size as organelles and other material inside the cell is copied and prepared for cell division. S phase is when DNA is replicated. We've already covered DNA replication, so we're not going to get into that too much. Now, G2 phase is preparation for cell division, and then M phase is mitosis, which you most likely know of the phases of mitosis as prophase, metaphase, anaphase, and telophase. Now, within the cell cycle, there are checkpoints, molecular checkpoints at each location that determines whether or not a cell is going to go from G1 to S, or S to G2, or G2 to mitosis. And so if the cell passes those checkpoints, then the next phase is entered. If for some reason the cell does not recognize needed material, or there's some sort of error in the cell cycle, what's going to end up happening is the cell should enter G0, which is a non-dividing state, which is what most of our cells are in at a given time. So the cell cycle is very tightly controlled. You can see on the bottom that it asks you, why is the cell cycle very tightly controlled? And it's because errors in the cell cycle can give rise to what we know as cancer. Now, chromosomes play an important role during the cell cycle. And so you can see from the picture that this is a picture of a duplicated chromosome. And so right here, what we end up with is actually what we call sister chromatids. And so sister chromatids are actually duplicated copies of chromosomes. The actual chromosome here is one singular piece of this. And so that classic X-shaped picture that we think about when we think about chromosomes, that's not entirely accurate. And sister chromatids are joined together in a central location called a centromere. Now, most eukaryotic cells have two copies of every chromosome. One of those copies comes from mom, and one of those copies comes from dad, and they form an attached identical pairs. So remember, chromatid is one member of the pair. Okay? And we'll talk a little bit more about that later on. Now, it's very important that we distinguish prokaryotic cell division from eukaryotic cell division. Remember, so prokaryotes only have one circular chromosome. So replication is much more simple. So the DNA replicates, and then you get a splitting of the cell. That DNA separates into opposite poles of the cell. Cytokinesis, or the splitting of the cytoplasm, happens, and then you get two new cells. And that is what is called binary fission. We can contrast that to eukaryotes because eukaryotes have many linear chromosomes. Now, you have to understand, and this is vitally important, you have to understand the chromosome-chromatid relationship. It can be confusing. So remember, one piece of DNA is the chromosome. Each chromosome contains a centromere. Now in S phase, when the DNA duplicates, we now have one chromosome but two sister chromatids. And then after mitosis, the sister chromatids separate and each cell gets a new chromosome. So a couple of the terms that we need to remember: haploid versus diploid. Haploid implies N, or one copy of every chromosome. Haploid for humans is 23, so our diploid number is 2N, or 23 times 2 for 46 total chromosomes. So if we have 46 total chromosomes, how many chromatids are present in G1? Well, we have 46 chromosomes, and so the DNA has not duplicated yet, so we have 46 chromatids. Now, if the DNA duplicates between G1 and G2, that means that we have 46 chromosomes. So how many chromatids would we have? We would have 92 chromatids. So once again, it's important, and that's going to carry out all the way through prophase, metaphase, anaphase, and telophase as 46 chromosomes or 92 chromatids until cytokinesis and the cell splits, and then each cell contains 46 chromosomes or 46 chromatids. Now, mitosis is considered non-reductive eukaryotic cell division. And by non-reductive, it means that we start with 46 chromosomes and the new cells end with 46 chromosomes. Now, here is something that I want to share with you about the phases of mitosis. AP College Board says that you do not need to know the names of the phases. However, I do think that it's important that you know the phases, the phase names particularly, because it's going to help you remember what's occurring. Since you don't have to know the names, you have to know what's occurring during each phase. So let's dive in and let's look at what the phases of mitosis are. So starts off with interphase. Interphase is G1, S, and G2. So this is when the cell is going to grow, particularly during G1, replication of DNA during S phase, and preparation for division during the G2 phase. And like I said earlier, this is about 90% of a cell's life cycle spent in interphase, or the non-dividing state. Here's a couple of images of what interphase might look like. Take note here that you don't particularly see the DNA, and I'm going to get into that in a minute. During prophase, the chromosomes condense because the nuclear envelope breaks down. So it is during prophase that we are able to see the genetic material within the nucleus of a cell. So something called a mitotic spindle begins to form within the cell as the nucleus breaks down. And in the animal cells, these organelles called the centrioles end up dividing. And so you can take a look at the pictures at the bottom of the screen, and you can see that you get kind of the breakdown of that classic nucleus shape, and that you can start to see the chromatin condensing. There's kind of a phase between prophase and metaphase called prometaphase, and so the chromosomes become much more visible and they start to migrate towards the cellular equator. And so you get two complete spindles that form at the cell poles. And so you can see a spindle here, and you can see a spindle here. And those spindles are going to play an important role in particularly metaphase and anaphase. During metaphase, the chromosomes are at the metaphase plate, or the center portion of the cell. So if you take a look at this picture, you can see that I'm kind of drawing where the cellular equator would be. You can see that the chromosomes within that cell are pretty much lined up at the metaphase plate, and the spindle attaches to the kinetochore of the chromosomes at the centromere. The kinetochore is a protein at the centromere that accepts the little spindle fiber created by the centriole. So what this is going to allow is, in the next phase, this is going to allow the chromosomes to actually be pulled apart. Now, following metaphase, you end up with anaphase. And so in anaphase, the kinetochore, those spindle fibers, are going to pull apart the chromosomes or the chromatids to opposite parts of the cell. And so the chromatids migrate to the cell poles mediated by the kinetochore. And so this is very much distinguishable when you're looking at an image in a microscope, because you will see the chromatids at opposite sides of the cells, or opposite poles of the cells. And lastly, you have telophase. Now, in telophase, what's going to end up happening is the chromosomes decondense because the nuclear envelope is going to reform. So you're starting to get that more classic nucleus shape. So the chromosomes are not as visible. And cytokinesis is going to take place, which is the splitting of the cytoplasm. So the cellular membrane or the cytoplasm splits, and this little area where the cytoplasm is starting to split right here is going to be where the cell divides, and that is called the cleavage furrow. Now, there are differences between plant-like and animal-like cells because plant cells have a cell wall, which means that that cell wall prevents that cleavage furrow from really forming. So when you look at in animal cells, that contractile ring of microfilaments pinches the cell in two. So you kind of get this pinching of the cell until it splits off, and now you have two new cells. Well, in a plant cell, you get what's called a cell plate that forms between the two daughter cells. That cell plate is going to grow until it pinches off between the two cells, and now you get two new plant cells. And during this time, organelles are being apportioned between the two cells, and it's completely random. One might get 100 mitochondria, another one might get 105. It doesn't necessarily have to be equal. So mitosis at a glance, if we look at it, let me go back real quick. If we look at mitosis at a glance, again, you don't have to know the names. You just have to know what's going on. For me, myself, the names actually help, because if I think metaphase M, chromosomes in the middle, anaphase A, chromosomes away. That's kind of how I always remembered it. You don't have to do that. Now, let's play spot the phases real quick. So if we're looking at this slide image of plant cells, and another plant cells based off the shape, if I look, this cell, I don't see any nuclear material, or I don't see any nucleus material. I don't see any genetic material. So cells like these are going to be in interphase. But here, on this cell, I see that there is condensed genetic material at opposite poles of the cell. So this is actually in anaphase right here. And if I look, same thing. I look here, this is anaphase. I look here, this is anaphase. Down here at the bottom, in the middle, I look at here, I see that these are lined up at the metaphase plate or in the middle of the cell. So this would be a metaphase cell. So you can kind of go through and you can look at these and you can spot and identify what phase are each of these cells in, and that's going to help kind of determine. You can look at this and say, well, the vast majority of these cells are in interphase because I can't see any of the classic mitosis steps occurring in these cells. So that's going to be the end of the presentation for cell cycle. I'm going to leave this up here for just a second so that you can look at it. Things that we need to know, of particular importance, describe the roles that mitosis plays. So what are the roles? We discussed that at the beginning. How does mitosis produce two genetically identical cells? How does interphase prepare the cell for mitosis? Why might many cells never divide? Explain the function of each stage. Notice I didn't say explain the function and name each stage. And compare the events of mitosis in plant-like versus animal-like cells. So that's going to be it for the cell cycle. Hope you enjoyed the video, and I hope that the video helps you out understanding a little bit about the cell cycle and mitosis.